The AACR Project GENIE is essentially, abbreviated as GENIE because it's Genomics Evidence Neoplasia Information Exchange. So this is a large consortium of global open access cancer registry that aggregates and harmonizes real-world clinical-grade genomic and clinical data from patients treated at leading cancer centers. About 19 centers all around the world have taken part in this consortium. This was launched in 2015 by the American Association for Cancer Research, AACR, and the initiative aims at accelerating precision oncology by providing a shared, collaborative, high-quality data resource for researchers, clinicians, and drug developers worldwide. And why this would be important in metaplastic breast cancer is we know that this is a very rare subvariant of breast cancer, less than 1% incidence. So even if you look at it institutionally, you're not going to have robust data to understand the genomic landscape, so to speak. So the pooling of all the data from worldwide helps us, understand in a larger, broad landscape of what the genomic profiles of metaplastic is about. So, these days, biomarker-driven therapy has really revolutionized cancer management. We have so many agnostic, approvals around. And so the evolution in the molecular profiling has opened up an almost an histology independent field with, these shared biomarkers. And that's one of the main reasons of why, we decided to do this project and look into the genomic landscape of metaplastic breast cancer. So the objective was to delineate the somatic landscape using the AACR GENIE version 16.1 data and to identify and see if there are any actionable alterations. So what we did was we accessed the CBio portal on website online. We found 267 samples, of which 258 patients were classified as metaplastic breast cancer. We looked at the different genomic profiles, including single nucleotide variants, indels, copy number amplifications, the mutual exclusivity, and also some metadata on the demographics, including age, sex, and race. When it came to demographics, the median age was 59 years, anywhere from 25 to 87 years of the cohort range. The cohort predominantly exhibited a female composition with 249 patients with 96.5%. There were 4 male patients in this as well. With race and ethnicity distribution, the majority of the patients were white, around 62%, and the others was unknown. About 25% were not classified. Asian distribution was around 6.6%, and African American distribution was around 5.58%, and Hispanic around 5%, and Native Americans at 0.8%. So very healthy, robust, representation from the different cohorts of race and ethnicity that was seen in this demographics. Some of the frequently altered genes, which is the meat of the study, was that, mutations in TP53 gene were the most prevalent and identified in 57% of cases. More than 50% of these patients did have TP53 mutations. The next driver was PIK3CA alterations, which was seen in 34% of tumors. And hence, these two pathways seem to be the most predominant ones. The others that we did see was the KMT2C and KMT2D, which are chromatin remodeling genes. And this also came close to 28%, suggesting that probably metaplastic cancers have an epigenetic dysregulation as a critical feature of their disease biology itself. Then the other most common mutation that we did see was FAT1, which was around 13%. And this actually implicates perturbations in the cell adhesion pathway. And we know that metaplastic, cancer does, operate through the epithelial mesenchymal transition. And this could be one of the reasons why it tends to drive through this pathway, so to speak. When it came to mutation hotspots, what we noticed was that, missense mutations in both the TP53 and the PIK3CA genes were the most common, although there was a significant minority in nonsense and frame shift and in frame deletion as well. In terms of other specific mutation hotspots in TP53, the most common one was R248Q, which was around 14.5%. And this is a well-known DNA binding domain hotspot that's linked to impaired, tumor suppressive function. Another subsequent mutation that we saw was R175H, which was at 8%, Q192 at 4.8%, H193R at 4.8%, and R273C. So all this kind of implicates that, when the key structural residues of the TP53 gene is aberrated, this actually leads to TP53 inactivation and hence causing that aggressive tumor biology with metaplastic breast cancer. So PIK3CA mutations itself, the most common mutation hotspot that we saw was the H1047R, which was at 55.1%. This was the most dominant mutation, which we know is well implicated in different other solid tumors as well. And other mutations did include E545K, Q546R, E542K, and C402R. And these mostly affect the helical and the kinase domains, but anywhere between 1 to 14.6% incidence was seen with these mutations as well. So all this implicates that, this PIK3CA could be a potential therapeutic target in terms of targeting metaplastic breast cancer itself. When it came to copy number alterations, we saw mostly in AGOP2, RAD21, which is involved in your DNA repair and RNA silencing pathways, RECQL4, which was at 20%, and MYC amplification was also seen, which kind of implicates that there could be some drivers of genomic instability and cell proliferation. There was also some deletions in CDKN2A and CDKN2B. And this essentially tells us that these key suppressors, tumor suppressor genes, which regulate cell cycle control, were also, aberrated. But predominantly, 66.1% of these patients had deletions, while 33.9% of these patients had amplifications. The interesting part was we not only looked at the AACR GENIE database, but we decided to look internally at the MD Anderson and database with collaboration with Boston Gene Genomics. And here we decided to briefly look at a cross comparison and a summary of the main genetic characteristics of metaplastic patients that were analyzed by Boston Gene. So although we had a very limited sample size, we had 4 patients who were metaplastic and who had undergone deep next generation somatic solid tumor tissue sequencing. And some of the pathways that we looked at was the PIK3, AKT, VNT, beta-catenin, the RTK and MAPK pathways. And about 504 genes were analyzed. And in this, among 341 breast cancer patients, we found 4 of these metaplastic patients, which amounted to 1% of the cases. And when we looked at the PAM50 subtypes, what we noticed is that there was one case with which looked more basal in nature, but the rest of the other 3 cases, these tumors actually behaved more like luminal A type breast cancer patients. So when we looked at their oncoprint itself and what the genomic drivers, really looked like, what we noticed was that it was characterized by profound TP53 inactivation. There was PTEN loss and actually EGFR pathway activation. And there was very low reliance on these canonical luminal drivers like PIK3, CA, and ERBB2, which made sense, especially when correlating with what we found on the AACR GENIE database as well. And what we noticed was that also there were some patients who had MET amplification, which also, potentially could be a therapeutic target down the line. When we looked at, like a multi-parametric box plot analysis of, the different gene expressions across the different, cancer subtypes, in metaplastic itself, what was very interesting for us to see was that there was elevated EGFR expression and there was a significant downregulation of CDH1. So hence, this really tells us that, these tumors really derive from a very mesenchymal and epithelial to mesenchymal transition driven biology. itself. But there was very low expression of hormone receptors like ESR1, PGR, the ERBB2, ERBB3, and hence, this really translated to its phenotype is mostly triple negative itself. There was some modest PD-1 expression that we did see, so there could be potential to harness immune checkpoint inhibitors in these subtypes, but there also was, there's a question that this could be a variable response itself. So overall, from this work, some of the translational insights that we were able to deduce is that, TP53 mutations, comprise more than 50% of these patients in the AACR GENIE database itself. So it essentially tells us that, metaplastic breast cancers really, are operating from a paradigm of genomic instability, impaired DNA damage response, and hence, these hotspot mutations probably benefit from synthetic lethality strategies by combining with other DDR, agents as rational therapeutic directions. PIK3CA alterations also are a very clear and actionable pathway, but because one thing that we did notice is that the TP53 mutations and PIK3 mutations were actually very mutually exclusive with statistical significance with a P of 0.043. So what this tells us is, there's probably a distinct molecular subgroups of these metaplastic breast cancers, so which probably benefit from a differentiated therapeutic strategy. So hence risk stratifying even the subgroups of metaplastic breast cancer based on their genomic profiles would be important when it comes to therapeutical success. And because we did see some chromatin remodeling gene alterations, definitely employing agents like EZH2 inhibitors and HDAC inhibitors, which are epigenetic modulators, would be very useful to test this preclinically and also for clinical studies. And also there were some frequent deletions in CDK into A and B and amplifications in oncogenes like MYC and RECQL4. So all this tells us that, if we are able to control the proliferator and cell cycle dysregulation, then we could have a compelling, translational avenue in terms of harnessing, therapeutic control in these type of tumors as well. So in summary, what we have demonstrated through this, large-scale genomic study is that, metaplastic breast cancer demonstrate a dominant TP53 mutated profile with frequent PIK3CA alterations, and hence there could be very distinct, therapeutic vulnerabilities because of this. So harnessing the genomic instability pathway, the DDR pathway, the PIK3A pathway, and your chromatin remodeling defect pathway could be, potential therapeutic targets that could be harnessed in terms of, mitigating the therapeutic, responses we've not been able to achieve so far, so far through all cytotoxic chemotherapy and even with immunotherapy. So some of the future directions that I think that we can, allude to and hopefully harness, using this project and the research that we've been able to, derive from is that I do think molecular subtyping of the different subhistologies of metaplastic breast cancer would be very important. So a genomic, precision therapy selection would be more useful for these patients to hence kind of improve their survival outcomes based on their genomic behavior. We also need to look into clinical trial development and, incorporating these patients more into basket studies, testing DNA repair inhibitors, epigenetic agents, PIK3 and AKT inhibitors.